Good morning, good afternoon. Thank you very much for having me in this very exciting webinar. I'm come from the University of Milan, the Center for Complexity and Biosystems. That was founded with some other colleagues with different disciplines in 2015. I'm working in a complex system, in particular on digital health, and I was training as a general pathologist, but I'm also actually is about more than 18 years that I'm working in digital health using different strategies to do quantitative biology and prediction. So what's the big question that I really, spend a lot of time of my research thinking about? Spreading of metastases. We know that now the people are not dying for primary tumors, but for metastases. It would be very important to predict in specific patients, what will happen to that specific patients in his or her future, where the metastasis will spread, and other questions like this. Why metastasis is the big problem? First of all, because most of the time the metastatic cells are in inaccessible sites. And second, because of the aggressiveness of metastatic tumor cells. Actually, we worked very extensively on both of these questions, and I will show you some data and results that we got in the recent years. Starting with the first point, spread of the cells in inaccessible sites. We know very well that metastatic process is a multi-step process, and each of these steps should occur in order to have, to give rise a secondary tumors and to see, a metastasis. What we did, since the cells can spread in inaccessible sites, and the question was, are we able to quantify and to predict in which organs, in which sites the tumor cells, the primary tumor cells will spread. So, and this is, of course, depending by two different aspects that can play together or not, the famous seed and soil. So is the structures of the blood vessels, lymphatic vessels. In this case, we focused on blood vessels. So the physics that is behind the spreading. And on the other hand, the characteristic of the cells, the aggressiveness of the cells, the adhesion capacity of the cells. So all the other issues that are, of course, involved in this seed and soil. So the environment, or the seed, the structures of the blood, of the circulation. What we did is, as I show you now in this video, we did a 3D simulation. So we reconstruct here. I'm showing the pancreatic cancer, but is happening. So we did the same for many different primary tumors. So this is just an example. So we did a 3D reconstruction of the circulatory system using MRI images. So 3D images. So I mean, we completely reconstruct the structures, completely, means that we have now a humanoid that recapitulate a human structures. And also, you can see now, you can appreciate this part. And we also recapitulate the blood flow models. So the model simulates hemodynamic linear blood flow equations from veins that you can see in red. Sorry, it's the opposite. Veins are in blue and arteries are in red. So we recapitulated actually the structures and also the hemodynamic laminar blood flow. So completely the physics of the blood flow system. Regarding to the adhesion properties, they were not. They were always the same. We didn't change them. So, and now you can see the primary tumors. These are pancreatic primary tumors that are spreading and going around. So this is circulating tumor cells that are released from the pancreatic tumor primary sites, and they are, you know, going whatever they can in these branching points. That mean turn and go randomly in the branching points. And so we simulated actually, the capability, the possible capability and the projection of the tumor cells. And you see here what, they are moving and going around according to the physics actually that is behind. And then what we did is, of course, we obtained some specific sites, metastatic sites, where these cells were more frequently, reached. So they reached the more frequently specific secondary sites. And what we did is to compare our data, the simulating data, with the autopsies. So we got a large data set with a lot of autopsies got from patients, from subjects that died for specific primary tumor. So the primary tumor was known, and then there was an autopsy, and it was counted in which sites they give rise metastatic metastases actually. And then you can see here a cross correlation, the comparison between our the simulation and the autoptic, the sites that was found in the real life, in real data. And you can see that we got different results according to the tumors. For lung cancer, there is a pretty well correlation, very good correlation between the simulation and the data. That means that the blood flow and the structures of all the systems, the blood flow system is very is very important for the spreading of these tumors for lung, but not for liver perhaps, and also for other kind of tumors are less correlated. And that means that in this case you can appreciate also better in these in these histograms. You see lung 82%. So I mean, this is geometry and flow, and in the red, actually in the dark pink, in the light pink you see seed and soil. So you see that lung, so the geometry and the flow is very important actually, and is matched very well what really happens in the real life, while for liver no perhaps, and also for other kind of tumors, but in particular liver and kidney. So that means in that case is very important, this specific kind of tumors is very, very important, the adhesion properties, the aggressiveness of the tumor cells, so all the other characteristic and capability in the mechanism that the cells use to give rise to metastasis. But for lung, geometry and flow is important, is very important, and can be used for predict in how many cells will give rise metastasis starting from that specific primary tumors. And so now we have a predicted models that for some tumors works well, and we can predict, given rise the circulating tumor cells, we can predict in which metastatic sites the tumors will give rise a metastasis and will growth, and that is a predicted model. But on the other hand, we have seen that for other kind of tumors, the aggressiveness is due and involves also the environment and the capability of the cells, probably the adhesion properties and other biological mechanisms. So this is the second point. We worked very hard, try to understand and to answer this question, aggressiveness of metastatic tumor cells. And in this case, we investigate a lot the epithelial mesenchymal transition. This is a key point of the cells. We have seen also in one of the cartoons at the beginning where I shown the the multi-step process of metastasis. Actually, the epithelial mesenchymal state means that the cells can change their state from a polygonal, columnar shape, so specific shape with apico-basolateral polarization. There is a strong cell adhesion, and they are not able to migrate so much, and they express some specific markers like Caterina. But they can, exactly by the environment, many different aspects, they can change, and they can go in the mesenchymal state. In this case, you can see that they change completely the shape. They are spindle shaped, they have these focal adhesion contacts, and they acquire the strong migratory potential. And of course, in this case, we have different markers. So this process is, can go in one direction, but also on the other direction from mesenchymal to epithelial states. So it's bidirectional. So it's this process is called by different ways. So like phenotypic switching, so the cells can switch from one state to another according to the environment, according to many different aspects. But it's not so simple like this, because more recently was shown like this transition between epithelial to mesenchymal state is not just from one state to another one, so abrupt, but there are also intermediate states. The cells can go through intermediate states, and these intermediate states seems to be very, very important, because the intermediate states seems to give rise metastasis and give, show a much more aggressiveness even with respect to mesenchymal state. So this seems not, this seems, an important biological questions. And of course, the capability to go in one direction or in the other direction is depending by microenvironment, the tumor cell modification. So many aspects can be involved. So my group worked really a lot and still working a lot on phenotypic switching. We publish a lot, and we try to identify, actually we shown and we demonstrated how there is actually a small population inside the bulk of the tumors that we call maybe not properly cancer stem cells, because they resemble some typical biological features of stem cells, but under specific condition by the environment can switch. So from one state to another, so a cancer stem cells can switch to a cancer cells, but also a cancer cells can switch to a cancer stem cells under specific condition. And so we worked a lot regarding this phenotypic switching, this capability of the cells to switch the phenotype in order to understand in order to define and to understand the possible targets, which targets we can use to stop this process. This is pretty important, because when a cancer cells switch to a cancer stem cells, that's of course can be related to quiescence, can be related to more aggressiveness, maybe in the future if it's not now. And so to stop this process and to understand which are the key factors regulating the phenotypic switching is very, very important. And what we found, so there are many papers, you can have a look now. There is now another one that is just accepted and will be released very soon in 2022 or 2023 regarding this process. But I can tell you that one of the key aspects that we demonstrated, in particular, my group worked a lot on melanoma. So in this kind of tumors, what we found is there is a complex network of macroRNAs that regulates this process, and in particular way, then we identified the macroRNA 222 that seems to be, at least in melanoma, pretty important for this process. Actually, we demonstrated that targeting this macroRNA, this phenotypic switching is impaired, so it's not going to work anymore in a good way. Anyway, you can have a look at the last and these papers if you are interested, but I'm not talking about this today. I don't want to talk too much about the days, but how we used, what we did for other stuff regarding the epithelial mesenchymal transition. So since the epithelial mesenchymal transition is a key factor, we also use an in silico approach. And what we did is to recapitulate in silico, again, the complex network regulating the epithelial mesenchymal transition. We started from Stanaway models that was released and published in 2015, but was not physiological because according to these in silico models, it was possible to go only on one direction, from E to M, but not vice versa. And as I showed you before, the process is reversible. So we added the other nodes, and now this network is reversible. So it's possible to go from EM or ME, and the strength of this network is that now I have the capability in vitro to knock out or overexpress one specific gene and see the release, what is the outcome. We published in 2018 on PNS, and what we got with this model, first of all, of course, we explore what happens in this model, starting from a random state, and then update the rule and find fixed points and see what was the outcome. And we found that we found two states, stable state, that was E and M state, as expected, of course, the epithelial and mesenchymal state, but between the two states, we found a lot of intermediate states, not few of them. And that is very important because, of course, as biologists, we are able to define intermediate states using some markers. So we see overexpression, so both, not overexpression, but co-expression of E and M markers in the same cells. But of course, we have a limited markers, and there is, we are, of course, limited when we do just in experiments, because we have a limited numbers of possible markers that we can use. But according to our models, so we can switch and on the different genes, and we found that actually there are many intermediate states, many ways that the cells use to go from one state to another one. And these states, and this is shown in this part of the pictures, display a high energy. What does it mean? That they are not stable and they are prone to move more, and so to give rise metastasis. This is actually what I've seen in experiments, that as I said before, the intermediate states are more metastatic than even the mesenchymal one. Then what we did is to map RNA-seq data on our landscape, and now we are able to map actually the RNA-seq data in our landscape. And you see that, as expected, this is a control, if I can say this. We are analyzing RNA-seq data from skin and fibroblast skin. All the RNA-seq goes data went on the epithelial state, while fibroblast in a mesenchymal, as expected. So that means that the model is not just a model, but works very well with the data. And we are now able to overlap, the RNA-seq data on the landscape. We also did these experiments. This is another control of our models. We started from cells that were in the epithelial state, and the cells were treated with some inducers to induce the transition from, in this case, from epithelial to mesenchymal state, and there were RNA-seq data, single cells at different time. And so we analyzed and we overlap, as I showed you before, the data on our landscape. And as you can see, we are able to follow the trajectory of the cells that are moving from the epithelial state through these intermediate states, and at the end arrives to a stable state that is a mesenchymal one. And we can do exactly the opposite, starting from, in this case, an M state, and they differentiate the cells with, in this case, with doxorubicin, and follow the trajectory. So this is very important. So we have now a strong instrument that can help us to follow the trajectory of the cells from the E and the M state, and using RNA-seq. We also analyze data, so we project this data on our landscape. This data were single cell transcriptome analysis of primary and metastatic tumors, so head and neck cancer. And what they found is there was this, again, intermediate states that was more aggressive, so they were metastatic, and they show, show these experiments was actually was not an experiment, but was data from patients. What we did again is to use this data and to project on our landscape, and what we found again is this the intermediate states, the patients that show these cells with intermediate states, and were also showing higher aggressiveness, where in our landscape were in the high energy state. So according to our our models, they were more prone to give rise metastasis, and was actually what was shown by the paper actually. So then we focus more, so this was actually in one sense the theory and the models. Now we have a fantastic model that can help us, and we can use to predict, and we can use to overlap RNA-seq data and to predict what happens in which states are the cells. And then we use this for breast cancer. But why? First of all, because we know that there are a lot of cases, and is the first cause of death for women. So I'm really, interested in this cancer for for many reasons. We know that there are 4 different subtypes of breast cancer, and actually this is a percentage, and in particular there is the subtype, the triple negative breast cancer, that represents about 20% of all breast cancer. But it's very interesting because there is no yet specific drugs. They are now under trials, some drugs that are released by different pharmaceutical companies. But since there is at the moment no specific drugs, most of the time the patients, as reported by the literature, are treated when maybe after was discovered that was not necessary, and the treatment was even worse than because of the side effect and other impact. So this is an important percentage, about 30%. So it's important when you have in this case scientific point of view a triple negative breast cancer, to predict in this subject what will be her future if that tumor is more aggressive or less aggressive, because it has an important impact on the therapeutic strategies that the oncologists can suggest to the patients. So we developed a platform using the EMT transition, and also using the other strategies that is at the base. There is also some artificial intelligence, for breast cancer prediction, and is the first platform that also is able to release to give a personalized strategy. You can see here the website where you can have more information technically, but I can just show you some data that we recently published using this platform to validate the platform for triple negative breast cancer. Here we published this on the Cell System in 2021. So here we analyzed patients with a diagnosis of triple negative breast cancer, and again we analyzed the RNA-seq using our platform, a little bit modified for breast cancer. And you see this is a single patient's RNA-seq, and you see that the patients are again are able, to stratified in different part of the landscape, and the more aggressive these red are mesenchymal one, the blue are epithelial one, and these actually were the patients with worse prognosis and more aggressiveness. Here you can appreciate better these single patients. You see here that we have here the relapse-free survival over time, and about 120 months. And you see that the patients that had a low risk according to our platform that we called Ariadne have a higher free relapse, so they were higher chance to survive with respect to this cohort of patients with the high risk score of Ariadne. So we have now an instrument, these platforms allow to calculate the risk, the probability that specific patients give rise metastasis over time, and can be used as a predictor. Then what we did is to compare our platform Ariadne with the tumor immune macroenvironment meta-signature, again for triple negative breast cancer. So is known that there is some possibility to stratify patients according to the immune system, to reaction of the immune systems. You see here there is some possibility to rank, to subdivide patients according to this immune signature, but there are also a lot of intermediate patients that we don't know actually, and the signature is not able to catch the specificity of that specific of that specific patients, so it's not working in that case. So then what we did is to use in these patients, in this data set, our platform Ariadne, and you can see that these intermediate patients that are not completely are not cached by this immunosignature, in this case Ariadne is able to identify a subset of patients with a high risk and high score of the platform. So again, can be useful even in addition with this immunosignature, in particular in this gray, in the gray part where the signature is not going to work very well. So this is the clinical practice for triple negative breast cancer of Ariadne. So it's possible now to use Ariadne for understand which patients can be used maybe for chemotherapy or for other treatments. And of course, what we want now to know is to see how works Ariadne with immunotherapy and targeted therapies. And we are very happy to to open also new collaboration. If you are interested, please contact me. We are very happy, to use our platform for your data and see what happens. These are the people involved in this project. Myself, Stefano Zapperi is a is a physicist, theoretical physicist, and Francesc He was our PhD postdoc for 5 or even 6 years. I don't remember, so a long time. And now he has an independent position. And we also, me and Stefano Zapperi, believe a lot in this multidisciplinary, interdisciplinary, new field in one sense, discipline. And we have written this Physics of Cancer that was published on Cambridge University Press, and I really, recommend you, if you want to, I don't know, if you come from biological discipline and you want to understand much more about the new quantitative strategies, or vice versa. Thank you so much for your attention, and I hope that was interesting for you.